everyone. So pleased that you could be here. And I'm Sandra Weiss. I'm one of the co-directors for the Depression Center in collaboration with my colleague Owen Volkovitz, who's the other co-director. And we are just thrilled to have with us for 2 days Jonathan Flint, and want to thank the National Network of Depression Centers for their funding of this opportunity to have him work with us. You may not know about the NNDC, or National Network, but it is an organization of about 28 universities now, each of which has a depression center, and the goals are to work together to try to enhance awareness of depression, research in depression, treatment, and education. And so our center here at UCSF is one of those. And I know that Dr. Flint's reputation probably precedes him, but I just want to say a couple of things about him before we begin. You may well know that he was really the first researcher in collaboration with Kenneth Kendler, who identified a robust kind of link, a genetic link with depression. And those findings were very seminal in terms of the other work that's been done since that time, with a very large population of women in China. And his current work is at UCLA, where he is now a professor in the Department of Psychiatry and Biobehavioral Sciences. And also he's with the Center for Neurobehavioral Genetics at UCLA. And they are involved in what's called the Depression Grand Challenge, which is probably the largest study that's ever been done to look at a whole mix of not only biological variables, but also social variables, cognitive, behavioral, environmental, a real multidisciplinary approach to understanding the origins of depression. So he is like the go-to guy when you want to understand depression, and is responsible for the genetics component as one of the leads in that whole effort at UCLA. So we're thrilled to have him with us, and the plan is to give about 10 minutes at the end for questions that you might have. So please welcome Dr. Flint.
Thank you, Sandra. So I'm delighted to be here. It's very kind of you to invite me. And I should be talking for the next 50 minutes or so about the work we've carried out on depression. And I'll start by giving a little bit of clinical introduction for those not familiar with the problem that we're addressing. Then I'll describe the study that we carried out to understand the genetic basis of depression, and finish with some observations that were rather unexpected to us and we don't fully understand yet about the role of mitochondrial DNA. So the fundamental issue here is that we don't understand what causes depression. And this is despite the fact that it's still one of the leading causes of morbidity in the world, that it contributes to the mortality. There's about 800,000 people every year committing suicide. There's 245 million prescriptions for antidepressants written, cost to this economy of about $10 billion. And even though, as most of us clinicians know, only about half of our patients show much response, and there have been no new or more effective antidepressants produced by the drug companies. So it's a big clinical need, and we think the only way that we're likely to change this is by understanding what causes depression. And our clue here is that there is, as in every other complex disease, a genetic component, and the problem is how do we actually get our hands on that genetic component so we can push forward the biology to help our patients. And I'm going to just give you next a very brief introduction to the nature of this problem, and I'm going to do so by using some data from a UK study. This is the UK Biobank. It's a study of 500,000 people. It's a very unusual study in that the people who generate the data make it totally available to you. Anyone in the audience who's got a anyone who's got a academic institution label can go in, download these data. And I'd strongly recommend you browse this website. It's an interesting place to go. And the slide I'm going to show you next is the application of these data to understanding the genetic origins of 3 diseases using genome-wide association. So for those of you not familiar, for reading these graphs, the horizontal axis, those numbers you can see are chromosomes, vertical axis is the likelihood of association, and the dotted line that you can see running in a horizontal position at about 8 or so on the vertical scale is a threshold, and anything that sticks above that threshold is regarded as significant. And you can see for Alzheimer's disease, coronary artery disease, and type 2 diabetes, there are many peaks indicating success, finding genetic localizations for these conditions, and therefore the start of an investigation of the underlying biology. And I've, of course, left depression blank so that I can give you the answer so that you can interpret success, otherwise in trying to take apart genetic disorders of major depression. So that raises, of course, the question, why has it been so difficult? And I'm going to go through 3 possible reasons. The first is, a lot of people have thought because it's a psychiatric disease, and all psychiatric diseases are difficult to study. And this is an easy argument now to knock down, and I've chosen this paper from Nature from last year, where the same genetic approaches have been applied to schizophrenia, and you can see from the title of that paper there's 108 loci. So just because something's genetic doesn't mean it won't work. A second explanation might be because it's not very heritable. So a lot of work has gone on in basic quantitative genetics to understand the heritability of psychiatric diseases, and most of this is using twin and family studies. And here is a summary by Ken Kender of what he regarded as the most valid, well-designed studies. And you can see at the bottom there's a summary, and the vertical line here gives you an indication of the heritability, which in this case is about 37%, and the horizontal bar shows you the confidence intervals, which is pretty tight around that limit. So it's a pretty good estimate, and indeed there's other work by Ken which backs this up, that the heritability is about 40%. So is that enough? Well, yes it is, because diabetes, type 2 diabetes, has a heritability similar to this, and I've already shown you the data for mapping type 2 diabetes, and it works. So that can't be the reason. So the last reason I'm going to put forward is because it's not one disease. And I'm going to show you 2 pieces of evidence that would support that, and one is based on heritabilities. So what I'm showing you here is, again, twin studies. One from, this is again Ken Kender's data, one from his cohorts in Virginia, and the other from a very large Swedish twin registry. And I'm showing you the heritability for males in red, heritability for females in blue. And noticeably, those are different. The heritability is significantly different. These are very large sample sizes. But the reason I'm showing you this is because of the green bars, which represents the genetic correlation between males and females. So if the same genetic variants made men and women depressed, that bar should be at 100%, and it clearly is not. It's more like 55, 60%. In other words, about 40% of the genetic variants are not shared between the 2 sexes, indicating that at that level you can regard it as not being the same, at least in a genetic sense, disease. And my second example is taken from the other major contributor to the causation of depression, which is the environment. And we're all familiar with the fact that a stressful life event disturbs our emotional state. And we know from a lot of work that there is a likelihood, if you have a stressful life event, that you will become depressed. And importantly, what that work tells us is there's a relatively well-defined window of about 3 months between having that event and the onset of depression. So in other words, if you come to me and tell me that you're very depressed, and I talk to you and it turns out that you had something unpleasant happen to you, you lost your job, your partner left you, and that happened more than 3 months ago, certainly it can happen 6 months or a year ago, then that's unlikely to be the cause of your sadness. However, there are other forms of lifetime events which don't behave like this. And the classic one here is abuse that happens in childhood, particularly childhood sexual abuse, a very, very potent predisposing factor for depression. And the interesting thing about this life event is that the consequences are not just around childhood, but they spread, as you can see, as I've illustrated in this picture, multiple episodes of depression throughout the lifespan. It doesn't have this relationship, this temporal relationship that we see at our onset events. Again, indicating that the pathways to the disorder are likely therefore to be different. So for those two reasons, and there are others in terms of the factor structure of the disorder, but those two reasons I think indicate that we should consider this disease as being heterogeneous in its origins. So working on that basis, I set up a study with my colleague Ken Kendler, and we designed it around that hypothesis. So we chose to work only with women. I've shown you the evidence for that. It's more heritable in women. And we also chose people who had multiple episodes of depression because of evidence indicating that the genetic effects are different. This is again from twin study work. I won't show you those results. And because there are many possible pathways to depression which we couldn't exclude in our recruitment strategy, we had to collect a lot of risk factors: personality, childhood sexual abuse, stressful life events, poor parenting, no social support, and a host of other features, particularly comorbid diseases like anxiety. These we had to assess and take detailed information to really include them in our modelling. Now, that meant that we needed two things. We needed a large enough population so that we could go in and restrict our attention to these specific groups. So although depression is very common, because we're only looking at one sex, we're looking at recurrent depression, we have age restrictions, other features which would reduce our recruitment. We needed to look in a large population, and then we needed to have a lot of their time, and we needed doctors who would be able to take that information. So that's one reason why we went to China. It's got a very big population, a lot of hospitals. And I went with Ken, and we would speak to the hospitals, we'd recruit doctors as interviewers, and then Ken and I would spend about a week. Ken has a long history in knowing how to train people to take interview history. And we give each of the doctors a laptop, which you can see there, and for about a week we'd have them rehearsing how to take interviews. In fact, for this study, eventually 80% of all of the interviews recorded, because we also had a set of doctors who were there as editors to listen to the interviews after they'd been taken and try and make sure that the data was of the highest quality. So there's a lot of quality control that went into this. And we started this in 2008, and the next graph describes about 4 years of my life where I was every week tracking the sample collection. And you'll notice if you look at these graphs that they're not following a simple linear trajectory. And what would happen is that there will be these vertical bars, you can see. And if you notice they're occurring fairly regularly throughout the period when we were collecting the samples. There are 2 weeks, these ones are the most marked. Those of you not familiar, which I wasn't when I started the study, in China, population of 1.5 billion, the entire country goes on holiday for 2 weeks. Nothing happens around New Year. There's also a week's holiday in October, the national holiday. So these are periods of great stress for me, because you never know whether things would sort of start up again, but they did. And it took us 4 years to collect this sample, but we did indeed end up with the 12,000 people that we intended to collect. But in so doing, we had to approach, in the end, about 60 hospitals. This is an indication of the places we were collecting samples from the far north in Harbin, right down to the south in Shenzhen. And it requires a lot of negotiation, a lot of time just to visit the hospitals and try and make sure they were working with us, convince them that it was in their interest to do so. Some hospitals, of course, would turn us down, others would be welcoming. And as you can see, this is quite hard work. It's a lot of time spent talking to a lot of people entirely. So in 4 years, that's what I was doing. And at the end of this, we collected the 12,000 samples, and then we needed to subject them to genetic analysis. And in the far south of China, there was a place called BGI Shenzhen. Shenzhen is the name of the city. BGI, you might think, maybe at one stage stood for Beijing Genomics Institute, but Shenzhen is 3,000 miles away from Beijing, so clearly not a really relevant name for this. This is still is the world's largest DNA sequencing center. And when I gave the samples to the scientists there, that's what they wanted to do, to sequence them. But we had nowhere near enough money to sequence 12,000 people full coverage. So we took a rather unusual strategy, which was low coverage sequencing. So this slide shows you roughly the coverage that we were obtaining. So there's a median about 1.7x. Most people work with a minimum of 10x. 4x is regarded as low. We're down to less than 2x in most cases. So there's a huge computational challenge here. The sorts of data we get look something like this, where in many cases we have no data at all. Other cases we have data from just one chromosome, and occasionally we get both alleles. And what we have to do is to turn it into data look a bit like that at the top here, and that's an imputation problem. And solvable, but it took us about 18 months to 2 years to run through this. There's about 40 terabytes of sequence data. We had to generate the calls ourselves, and I remember at the end when we were doing some quality control and got a few genotype chits done on this data set, that the student who was running this analysis was astonished that we could generate in a few days what was been taking us months to weeks just with chips. So there's a lot of discussions went on as to whether I really made the right decision with the sequencing. But try and convince you that we got a lot of extra data out of this that we wouldn't otherwise have obtained. So having generated the data in that way, we then carry out a fairly standard genetic association. So I'll show you our results, and you should be able to now interpret this in the same way that when you looked at the figures I showed you from UK Biobank. And you can see that there are 2 places in the genome that exceed significance. That, of course, is not really enough to convince geneticists. We need replication data, and we were very fortunate to have a collaborator in Beijing, and I'll show you those data next. So let me just show you the results for the 2 loci on chromosome 10, where we had a genetic effect. So I'm showing you here the allele frequency, the odds ratio that we detected, and the last column here is the p-value. And our collaborators in Beijing had a sample size smaller than ours, not so well phenotyped, some issues about how comparable it was, but at these 2 loci we did find a significant effect, and the joint results of 17,000 people are shown on the far right. So we found something and we could replicate it, so we felt that was a reasonably robust result. Now, I want now to just go back to the question that I raised at the very beginning, which is why it's been so difficult. And remember, I said to you that I thought that if we treated this as a problem in heterogeneity, we'd be successful. So we have been successful in finding 2 loci. And of course, the question really arises is, was that due to the fact that we've excluded heterogeneity, or is there some other reason? We were just lucky. So I want to try and convince you that is true by just giving you 2 ways of looking at this data. And I'm going to look at this by categorizing our data in 2 ways. I'm going to subdivide the data by severity of illness and then in terms of the etiology. Now, severity of depression is not quite so straightforward. It could be, you might think, the number of episodes that people have had. That might be one measure. But then you can immediately realize that if people only have multiple episodes of 2 weeks, that might not be the same as someone who has a single episode lasting 2 years, which is more severe. It might be the number of symptoms people have got. Is that going to be a marker of severity? In the end, we decided to use a very old way of categorizing severity, a diagnosis called melancholia. One of the characteristics of this is a specific type of affect. The mood itself should be unusual in a way. And what we had in our sample was about 4,000 or so cases of melancholia. So let me just go through this by showing you, initially, that's what I showed you before, that's our initial results. And I'm going to show you the results from the 4,500 cases of melancholia. So we've noticed we're reducing the sample size almost by 10% here. What happens to the genetic effect? What would you predict? That is what we find. There's a change in the genetic signal. So one of those loci actually becomes more significant and one is a bit less significant. And our question then was, is that a significant change? That could just be a chance effect. It's struck us as unusual that we would see an increase in the signal, but we didn't know for certain how unusual. And the way we addressed that question was to randomly sample 4,500 cases from our total number and then rerun the analysis and see how often we got a signal that changed. So here is a summary where I'm showing you in blue the subsampling. On the horizontal scale is the odds ratio. And the red line is the result that we get from our real data. And we can see where that lies in that distribution, and as indicated on the right, we can actually assign a p-value, which is a 0.01. So a simple criteria that indeed is less than we'd expect to have happened by chance. And the second way I want to show you that there's changes in the heterogeneity is by looking at the way that we could subdivide by our environmental causes. Now remember I said that there are a number of environmental risk factors, and I pointed out to you that childhood sexual abuse is a causal risk factor, a major one, and I'm showing you the data from our study. We divided up the risk as non-genital, genital, and intercourse, and you can see from the odds ratios shown in the middle there that as you increase the severity of the abuse, you increase the odds ratios, consistent with this being a causal factor. These are very similar to data from other people's work. It's a very big effect, the single biggest effect, that, as I said, that's been reported for this condition. And we also collected this information on stressful life events, and I'm showing you a list of the events that we found here. And if your eye runs across the odds ratio column on the right-hand side, you'll see there's heterogeneity there. And for example, a very severe effect of rape, less effect for some of these other conditions. Again, consistent as you might expect with the operation of a more severe effect having a greater risk factor than rape. Again, consistent with what we know from the literature. So these data are behaving as they should do. So we asked the question, what will we see in our genetic data if we remove subjects who experience stress? That's to say, let's take our full sample and ask, are the genetic effects similar in those who do and don't report stress? And we found the following result. So this is now reducing our sample. We've reduced it from over 10,000 to 7,000. And again, we're seeing a change in the genetic signal. We're seeing some low sign. Just to make this clear to you, the next slide, I'm showing you again our primary finding with 10,500 people. And this below is the sample size reduced to 7,000. And you can see that one of the loci on chromosome 10 seems to have shrunk out of existence. And we're getting new signals that were not apparent at all on chromosome 1, and a large, very large signal on chromosome 8. This was rather surprising to us. We didn't think it would be such a marked difference as this, but it seems to be the case. And we were able to replicate the signal on chromosome 8 by looking at the Psychiatric Genomics Consortium data. So I'm showing here what's called a forest plot. The Psychiatric Genomics Consortium is a large group of people collecting data from lots of different cohorts, which are listed here. And if you look, this is a summary. The black, the bold here is PGC1. This is a significant effect here. This is the Converge data. Converge is our sample. This is not having removed the environmental effects. This is just the main effect of the data and the p-value given down there. For the combined meta-analysis for the p-value 2.5 times 10 to the minus 7, if we put in our results from removing the environmental effect, that becomes far more significant. So we're pretty certain that locus is correct. Chromosome 1 locus, we can see an effect in the PGC data, but it doesn't reach even the 0.05 significance. It's at least in the same direction, so there's some support for that. And then the other thing we can do is ask the question I asked before, is this change in the genetic effect significant or not, by doing this subsampling? And the results are very similar to what I showed you before. Indeed, the changes we see are unlikely to be due to chance. And the last way that we looked at this was to ask, how does the heritability of depression change in the cases with and without stress? And this is from the data, from our own data. We estimate the heritability in the entire sample is 21.6%, and this increases in those who don't report stressful life events to about 26%. However, that is not a significant difference. The standard errors on that are still quite large. It's at least consistent with the hypothesis that we're seeing different genetic contributions to these conditions. In other words, we have evidence in our data that there's heterogeneity there. So I want to move away now from the genetic data and look a little bit about what might be the underlying biology. So I'm going to start by showing you a zoom in for the three regions on chromosome 1, 8 and 10 where we're seeing genetic effects. And well, it's a little hard to turn these directly into genes. Always a risk. We're finding positions in the genome that contribute to risk. We're not finding genes. But as a first approximation, many people do this, you can look at, well, what's the nearest gene? So let's just take that. And what we found, these are the three genes here, what we found was something just slightly unusual, which is these are not genes involved in neurotransmitter release, not genes in neurotransmitter function at all. They're involved in mitochondrial function. So one well known to the metabolism community, NAD class, acetylase, an LPGAP1 localized to the inner mitochondrial membrane, and this transporter, mitoferrin. And the reason this caught our interest is because we'd already made another observation, which we were struggling with. The mitochondrion, pictured here as an EM, contains its own genome. So when you sequence DNA from a person, you sequence their nuclear genome, and you also sequence the genome of this organelle. Most people throw this out. We, however, noticed something which we didn't understand at the time, and that is that the cases in our study had more mitochondrial DNA than the controls. I'll show you that result next. So I'm comparing cases and controls. This is a normalized amount of mitochondrial DNA. Have to take into account the fact that, of course, we've got different amounts of nuclear DNA, so we control for that and normalize it. And this is not a big difference, but of course we have a very large sample, so it's highly significant. And we'd seen this result before we got the genetic data, and of course thought this must be some sort of artifact. And there were a number of things that ran through our heads when we saw this. Might it be a drug effect? Could it be a change in the cellular composition? And some other compound that might explain this. All of our patients are taken out of hospitals, so a lot of them are taking antidepressant drugs. Maybe it was just a side effect of the medication. So we tried to reproduce this effect, and we did so by giving antidepressants to lots of mice. And we gave it to a period of time, so I'm showing you results from taking mitochondrial DNA assayed in saliva. The reason we looked in saliva is because our DNA sample came from saliva. Taking saliva from mice, not easy. It took us a while to work out how to do that. And these are our fluoxetine and control, and there's really no difference that we could see. So we couldn't reproduce that. We tried another couple of other antidepressants, nothing really could reproduce the change in mitochondrial DNA. So it seemed to us unlikely it was that. What about a change in cellular composition? Maybe there's some subtle change that's been going on. It turns out that saliva, I didn't know this, is about 70% white cells. The other remaining is sort of fibroblasts, endothelial cells. And we worked a little bit with some immunologists and generated these data. What I'm showing you here are white cell components. These are the B cells, T cells, monocytes, natural killer cells. And we measured the amount of mitochondrial DNA in each of these. And you can see it varies substantially. This is a log scale here. So there's almost 100-fold difference between natural killer cells and CD3 cells in terms of the amount of mitochondrial DNA. So a small shift in the composition of the cells might have produced this effect. So we wondered how we could assay this. And the way we did it is by using methylation. So the methylation state of a tissue can be accessed from its DNA, and methylation is a relatively specific marker of the specific of cell types. You can say where a cell comes from. Show you the results we got. The way we did this is it's an expensive assay to do, so we decided to take the extremes of our distribution. We take samples with a very high mitochondrial DNA content and samples with a low mitochondrial DNA content. And what I'm showing you here are the methylation assays for white cells and for markers for endothelial cells, covering what we thought was a good spectrum of what the cell types were. And these are the cases, on the right are the controls. And as your eye goes over that, you can see there are some differences. There's a little bit more blue here than there is there. So in order to test that, what we did is simply regress out the effects of methylation. And on this graph, I'm showing you the main effects, not taking into account the methylation differences. So there's a big difference here because we're taking out the extremes of the distribution. So you expect to see that. And we regress out, and this is the residuals of that analysis, and it's still very significant. So we couldn't explain it just by changing the cellular composition. So maybe there's some other compound going on we didn't know about. Maybe something specific to our cell type. Maybe something to do our sample. So we approached a group in London who had a set of samples, and they gave us the DNA extracted from blood. This is a relatively small number, and we saw, again, a significant difference in the same way. So now we were becoming convinced that this was something more related to the biology of depression than we had initially taken for granted. And by this time also we were getting the genetic data which I've shown you, which indicated that maybe was a contribution from the nuclear genome, which involved mitochondrial genetic variants in increasing risk. We have one other clue, which I'll show you next. So what I'm showing you here is the amount of mitochondrial DNA plotted against the number of stressful life events. And what you can see is there's a small but indeed significant increase in the mitochondrial DNA with the number of stressful life events. So we wondered, therefore, was this relevant? And this graph shows you the total number of stressful life events in cases of controls. It's a very classic picture in that, as you'd expect, there was more stressful life events in cases than controls. But importantly, you find many cases report stressful life events, and many controls with stressful life events, and many cases who don't report any stressful life events. So we partitioned our data in the following way. Here are the number of stressful life events. I'm just giving you 0, 1, and 2. This is the controls and cases. And then we just look at the amount of mitochondrial DNA and ask us if there's a difference. And the important thing to bear in mind is that people who reported no stressful life events still show a substantially significant, not large, difference. So that indicated that depression might be mediating the predicted power of stress. So we came up with the idea that possibly this was a state effect. It wasn't a sort of historical effect. So could we test this? And we've done so in a couple of ways. The first way was to ask whether we could reproduce this effect again in mice by stressing mice. We can't give them the chance of sexual abuse. We can abuse them in other ways. And we can stress the mice by shaking the cages, giving them a small electric foot shock, restraining them. And then we can ask what happens. And here are the results. This was carried out over a 4-week period. And you can see that there's a gradual increase in the amount of mitochondrial DNA over time. This is not acute. It's really only apparent with a chronic application. And there's a gap here between 4 and 8 when we stop the experiment to see what would happen if we rested the animals. And this indicates that it's at least one component of it is dynamic. It returns to not significantly different from controls, but we don't know whether there's a residual effect there. I think that might be an important part of this.
This is mitochondrial DNA in what?
So we did it in lots of different tissues. This is in fact from blood. But we did it from brain, liver, muscle, that's from the ovary, and we did it from saliva and in blood. We tested all different sorts. And we see the increase in blood, and we see an increase in saliva, we see an increase in liver, We don't see an increase in brain. There's a bit of a decrease, if anything, in not significant, so in the other tissues there. We've also looked in sub-brain regions. We weren't able to do this in mice. It's easier to work in a slightly larger brain. So when we did it in rat brains, we found hippocampus and amygdala known to be areas involved in regulating emotion. We do indeed get changes consistent with what we see in the external tissues. So it's mixed across different tissues. So that experiment suggested that stress itself was driving this effect. But of course,
was there a direct correlation in the mtDNA between hippocampus and amygdala with saliva or blood? Were they related?
So it goes up in the amygdala and it goes up in the hippocampus and it goes up in saliva in the same direction.
In parallel with the same animals?
Well, we don't know because we've only got we only have one time point in the rats. So we haven't done this on a sequential basis. We don't know the answer to that. The other issue is, of course, what is happening in humans. And we didn't have longitudinal data. We couldn't access that. So we haven't designed our study to answer this question. But fortunately, we had some collaborators from Germany who had a study set up where they were bringing in patients with depression into a hospital and following them up for 8 weeks with a treatment regime. And they'd taken blood samples every week for these cases and controls. And this is what they showed us, and I was a little surprised at this. So time naught here is actually when the patients are admitted to hospital, when their depression is worst. And as they come under treatment, then the symptoms resolve. So we're seeing a time course here. I thought we'd see something that started here and dropped down, but that's not what we're seeing. So we're seeing it actually increase and then gradually coming down. And it's not actually back down to the steady state, the control level, even after 8 weeks of treatment. This raises a number of issues. First is just how does this relate to the phenotypic scores? So what they've got is a standard measure of depression, the Hamilton, it's a questionnaire-based, rater-based measure. And this is their scores, and you can see that, as expected, the people who are coming into hospital have high scores, and as they go through the treatment regime, it drops off. Controls, of course, actually at 0, as you'd expect. So that's perfectly expected. Just to show you this in comparison with the mitochondrial data, so that's the phenotypic data, the mitochondrial data. They're almost inverse here. What I think is going on is that this line is probably going to go and decay over time, but we don't know that because they stopped the study at that stage. We don't have those data. And it did raise the interesting possibility that although at this stage people are saying that they're feeling well and their symptoms have resolved, biologically they're not well. And therefore, would it be a more appropriate way of treating people is to keep them on the antidepressants and use this as the measure for recovery rather than what they're telling you? Again, we don't know the answer to that, but it's intriguing. The last thing I want to tell you is how these findings might relate to the causes of depression. All of this tells us this is some state marker, but I told you that we have some evidence from looking at where the genes are that contribute to the risk that it might be mitochondrial genes. So the first thing we work out is the genetic correlation between whatever is causing the increase in mitochondrial DNA and the increase in risk for depression. And that correlation I've written down here is about 0.4, which suggested that there is some shared genetic correlation. So we did the obvious thing, at least for geneticists, which was we map the amount of mitochondrial DNA. We got a very nice result. Very high. It's very nice working with the molecular markers. It's a much better thing to work with than depression. Very clear results. And when we looked at what these genes were, it reassured us that we're getting the right thing. So the biggest effect was over this gene here. It's right in the 5' region. It's gene TFAM, transcription factor for mitochondria. It's well known to be involved in regulating the amount of DNA. So that tells us that That's pretty cool. This is CDK6. It's a site-independent kinase involved in cell division. And there's a marker in an intron of it. Maybe it's involved, maybe not. It's not known to be involved, but it's a reasonable place to start. But neither of these genes makes any, or neither of these markers makes any contribution to the risk for depression, which is a little bit disappointing. And we've not been able to find anything else from that scan which told us whether there was a molecular contribution to the risk of depression. But there was one thing that did. This is the last piece of data I'm going to show you. So remember, our coverage for nuclear genomes is about 1.7x, low coverage. But each cell has hundreds, sometimes thousands of copies of the mitochondrial DNA. So when you generate low coverage sequence data for the genome, you have very high coverage for the mitochondrial genome. And this picture is showing you the sequence reads that we generated across the mitochondrial genome. Each of these grey bars is a single sequence read. And each of these little coloured marks is a position in the genome which is different from the consensus sequence down here. Now remember that there's only one mitochondrial genome. You just get it from your mother, so heterozygosity is not present in the same way for the nuclear genome. But there are places in the genome where you get a difference, a consistent difference, and that's called homoplasmy. It's indicated here. And there are also places, and you can see here, I'm picking up blue dot, which is consistent across a number of different reads, indicating heteroplasmy. So there seems to be some variation in the amount of sequence, in the sequence structure of some of these reads, heteroplasmy. Not clearly, even with a high coverage here, if we've got, say, something with 10% heteroplasmy, our ability to detect it is limited. So we don't have great power to pick up these effects. What we found was no correlation between the homoplasmic sites, no correlation between any individual heteroplasmic site. But when we added up all of the heteroplasmic sites, then we found that there was indeed a significant effect. So there seems to be an increase in the amount of heteroplasmy. And we were able to reproduce that in our stressed mice. We looked at the sequence from those and indeed found that to show the same thing. We don't know what that means. There's something going on here that we still don't really understand it. So I'm going to leave that there. We can come back to that in questions, and I'll just summarize what we've found. So two replicated loci that contribute to the risk in the main sample. I've shown you that there's evidence from genetic analysis that you can see phenotypic heterogeneity in our data. The amount of mitochondrial DNA is associated with depression, and we think that's in response to stress. And we show increased rates of heteroplasmy in response to stress as well. I'd just like to thank all of the people, particularly the Chinese hospitals and BGI, for this work. Thank you very much.
So time for questions.
I was just, thank you for the excellent talk. I was wondering, going back, when you originally decided on how to pick the severity of depression, what was the logic going through your mind where you then decided to pick cholia?
Really from the literature. So there's a very long discussion in the literature as to whether melancholia is a subtype of depression or not. And there's some quite heated opinions that maybe it ought to be a separate diagnosis altogether. He has different features. And those features focus around many of the components that I think a lot of us would think of as being severity. So that's really why we took that. There are, as I said, if we take other features, you can always argue. And one of the problems with going with many of the clinical features is just how robust are they. And we've got data where we've compared what previous interviewers have taken, so we just look in the medical records. And as others have found, things like number of episodes, length of episodes are not very robust measures. There's a lot of noise in those. So this has a background of literature to support it, and it gets us away from some of the issues of using a less robust clinical measure. That's why.
As far as the melancholia is concerned, it looks like most of your patients were melancholic. Is
yes,
that right?
indeed.
Yes. So one of the reasons that people may not I don't know that most studies have mostly melancholics.
No, they don't. This is very unusual. So we have about 80%.
Yeah.
We looked in other people's studies, and most people, maximum was about 15%. And it's begin. So there's an issue here as to, again, about ascertainment. So we've gone into hospitals in China,
Yeah.
where a country where admitting you've got a psychiatric illness is a big problem, and something like depression perhaps even more so. So you've really got to be ill to get into those hospitals.
Right, so the reason you may have succeeded is that, whereas others did not, is that you're studying different populations.
Indeed, that's so. And I think that's been a key to what's gone on. And another thing to bear in mind here is that when we designed this study, we'd included screens for alcohol, drugs, smoking. Population we're going for, 40 to 50-year-old Chinese women. They don't drink. They don't take drugs. They don't smoke. I don't think you could find anyone in the whole of California that fits into either of these.
In your treatment slide, the mitochondrial load at time 0 actually seemed to be lower in cases than controls.
Yes,
So,
not significantly, but yes, it was.
So how, if you just take that time 0 before treatment, how do you blend that with your overarching finding that it's higher?
I think the answer to it is given really by what we're saying here. So here, these animals already had this is the first week or so, this is by 2 weeks. We're not seeing any difference in the amount of mitochondrial DNA. If you were to go in and after a week where these animals have been kept awake, they've been having foot shot, they've been stuck in little tubes, and ask them how they're feeling, they'll tell you they're feeling pretty bad. But we wouldn't see a change in the mitochondrial DNA. So my guess is that what's happening here is this is fairly early on. This is not significant, this difference, so I'm not too concerned about the fact that it's lower. But I think that's the reason for this.
So you talked about the possibility that depression with a stressful life event could just be less heritable in general. But what about specific interactions? Do you look at like correlation in heritability or for potentially separate genetic effects?
Yes, we do. And I've been working back with Noah and Andy, who are sitting over there on this very question. We have some. So what we find is, so the first thing is that most people seem to work under the hypothesis that you'll see genetic effects coming out when people are stressed. So there's a standard sort of G by E view of the world, is that if you stress people, then certain genetic effects come up. We see the opposite, so that's unusual. We also see that if you just run a standard G by E test on that locus, then it comes out as being significant. And there's if you look at the two groups, the exposed and the non-exposed, if you like, they actually both have higher heritabilities than in the entire cohort, consistent with this being heterogeneous.
Really interesting talk, and I'm just thinking about the mitochondrial DNA stuff. And there's new research coming out that indicates that some of the mitochondrial variants that are in the brain are not actually represented in the peripheral blood. And so when you sample the peripheral blood, you may be missing variation in the brain. And the brain's obviously very interesting and unique in that its brain functions are regionalized. There's areal specification of the brain. And I'm wondering whether or not you guys have thought about sampling different regions of human brains to determine whether or not there's heteroplasmy across the cortex, like loci of the brains that correlate with mood symptoms.
Yes, we've thought about that. Don't have access to the samples. One of the surprising things is how rarely people collect information about stress. So, for example, so what we need is we need the brain tissue. What we've needed also with those with that measure. And of course, there's again an issue about what is the effect of death on this. So how much will we be picking up something that would reflect the cause of death and not actually be due to the stress that people have put through. So it's definitely worth asking. I'm not sure we can answer it with the available resources, but maybe with a big enough sample, we might see some effect.
When you measured or you sequenced the mitochondrial DNA, at what time point was it? At 0 time point? So the question
Do you
going
mean for
forward.
the mouse data or for the human data?
This one was,
So this is from our, we haven't got any sequence data on this group. This is from a separate cohort, which German collaborators have got, and they'd given us DNA from 0 to 4, 6, and 8 weeks after treatment.
so you don't know at this point whether or not the change in the quantity of mitochondrial DNA is reflected by the different sequences of the DNA that might occur.
We
So
don't
there's
have,
mutations.
so we don't, for this sample, no, we don't have any sequence data. And I don't know how, these data consistent with what we saw in the mouse suggest this is a very dynamic measure. It's going up and
Right,
down.
so it's possible if you have multiple types of mitochondrial DNA, those with mutations, those without, that one mitochondrion might replicate much more
Yes.
rapidly than others.
Yes.
And so,
So exactly, and what I didn't show you is that we had evidence that this is hard because the changes are small. So picking up this is a small amount, but it would make perfect sense that if you put the molecule under pressure, that it would accumulate. It's more likely to accumulate mutations in a position that increases its replication. Indeed, we have one place in the replication region, in position 513, which does indeed increase just about significantly, suggesting that that's what's going on. And ideally, what we'd like to be able to do is follow that region over time and see whether it goes up and down and understand that biology. Yeah. It's interesting.
Jonathan, someone over there is good.
Great talk. Certainly there are many adaptations in the body to acute stress that are helpful. And it could be that this is an adaptive change to stress that helps function. It wasn't correlated with depression, is that right? It wasn't linked to depression.
So it is correlated, but we think it reflects, as it were, the state of being depressed. So it's a state measure. The prediction would be that if you suffer some severe stress, you'll show this regardless of actually whether you are or aren't depressed. It should be some measure of stress, but we'd expect people who are in that degree of stress would report some affective disorder.
Are there any conditions that are known to be pathological where this increase occurs?
This is something very few people measure. It's generally chucked away. And there are few... So there's a couple of odd things here. So you may know that Parkinson's disease, the genes involved in Parkinson's disease are turning out to be involved in mitochondrial metabolism. So Parkin and actually in some of the biology that we're touching on here. And Parkinson's disease also turns out to present very frequently before motor disorders with a mood disorder, suggesting that there's again some causal link here. And it's an obvious thing to do is to ask, well, what happens to the amount of mitochondrial DNA in that condition? So I'd love to get my hands on some of those samples. And then I thought, well, just generally, what about even in schizophrenia? I mean, this is a pretty awful condition. People are under stress. What happens to the amount of mitochondrial DNA there? Again, no one's got the data on this. It seems to me there's quite a lot you could do. So how specific this is, I don't know, is the short answer. It's not been
When you're screening, do you also account for like anxiety? Like how does that play into?
We collect a lot of information, including 5 or so different diagnoses of anxiety. One thing we could do, which we haven't done, is subdivide our samples by that diagnostic category, just in the way that we've used the environmental data, the stressful life events, and look at the genetic effects across that. We haven't done that yet.
So, just a question, the odds ratios are still sort of fairly modest odds ratios.
You mean for the effects or risk for depression? Yeah,
Yes.
that's fine.
So you have a hardy sample size, relatively few targets that appear, and these very modest odds ratios.
Yeah.
So the question is a more global one about where does one go from here with genetic studies of depression?
I think there are a number of lessons. As always, you need a larger sample size. We're sort of just seeing a little tip of the iceberg. if you like. If we did a larger sample size, would this pattern of loci being involved in mitochondrial biogenesis be replicated, or will we begin to see parts of different biology emerging? The second lesson is that we've argued that going for a subgroup has increased our chances. We've tried to make it homogeneous. But we're also seeing that if we divide up our sample by these clinical criteria, we're seeing genetic effects operating differently. So should that therefore alter the way we design these studies? Would it be better for us to go for subgroups? So should we just target people who report no stressful life events against those who do? Would it be worth thinking about that? So I think those are issues which this study doesn't answer, but I think those are questions that we need to consider, and we are certainly so doing.
Are there regulatory elements in the mitochondrial DNA that are responsive to things like corticosteroids or things like that?
Whether that's, I don't think there's anything responsive to steroids. I didn't show you this, but we did inject our mice with steroids, and we could show an increase in the amount of mitochondrial DNA in response to that. But we don't know how that's mediated.
Yeah, it could be secondary.
Of course.
Yeah.
We don't know.
So it's a stress marker.
Yes, it's what it seems to be, is
Yeah.
a stress marker. And it comes back to this question of, is it a generic stress marker, or do you only see it in affective disorders?
Generic.
I suspect it's generic.
Yeah.
It would make much more sense if it was.
Sorry, just one more question. So regarding the sort of modest odds ratios, I mean, how much of the heritability do you think could be hiding in the sequence of the mitochondrial DNA itself, since that's also inherited?
A small amount is the short answer. I don't know the exact figure. But the other thing we have done is for the mitochondrial genome, 13 genes, trivial amount. But there are many other genes, about 1,000 or so, in the nuclear genome which are involved in mitochondrial function. And one thing we've done is try and ask, does that make a contribution to the heritability? And the answer to that question, that indeed is enriched. So we seem to see more of that come from the than you'd expect by chance. Again, consistent with the observation we're seeing from the loci that we picked up.
So unfortunately, okay, one more.
Thank you, Dr. Flint. It was a great lecture. Can you please quickly comment on the potential of the mitochondrial DNA as biomarker in comparison to telomere length?
We find that there's a negative correlation between telomere length and the amount of mitochondrial DNA. They overlap to some extent. Adding them together increases predictive power a bit, but not much. I think most of us regard a biomarker as something clinically useful, and I would be able to use this to change my treatment. They're not anywhere near predictive enough for that particular combination of patients.
Well, we want to thank you so very much. Hope that we can get it back again soon, right?